Welcome to this first video of Topic 3, Genetics for IB Biology. In this video, we'll be looking at section 3.1, where we focus on genes. So starting off looking at our understandings. In this section, we really need to remind ourselves of what a gene is, and we would have studied this type of biology in earlier years of our education. Okay, and the Australian curriculum, you would have studied this in year 9 and year 10. Okay, so let's refresh our memory of what a gene actually is. And we're remembering that in our nucleus, we have this chemical compound known as DNA. It's a very stringy, spaghetti-like substance. Okay, now DNA makes up these structures called chromosomes, and along those chromosomes are what we call genes, and they're the coding blocks for different features and characteristics of organisms. Okay, so we call our gene a heritable factor. It's something that's passed on between generations, and it codes for those specific characteristics. Now, we talked about chromosomes because chromosomes are made of sections of DNA, which we call genes. So chromosomes are made of DNA molecules. Each chromosome contains these smaller sections, which we call genes. Okay, so you can see in the diagram here, we've got our DNA, and you can see a section of it's highlighted in a creamy colour there, and that's our gene. And all that winds together around our histones to make our chromosomes. Now, each gene consists of sequences of hundreds and thousands of bases, just depending on which gene it is. So remembering our bases being our adenine, thymine, guanine and cytosine. And we'll remember that from topic 1. Okay, so where are genes found? So as I said, these genes are found on the chromosomes. Okay, now the actual point on the chromosome that the gene is found is the locus, or known as loci. Right, now we have these things called alleles. Now this is not really that confusing, but to start off with, it always seems to confuse students. We've got 2 chromosomes, and on each chromosome is the gene to code for a certain trait. So it could be hair colour, for example. Now, hair colour, there's different types of that gene. And the different types of the gene are what we call the alleles. So in the picture here, we've got the gene for flower colour in a specific plant. Okay, and you can see that one allele is for a purple flower, whilst the other allele is for the white flower. Most animals and plants have 2 copies of each chromosomes, so we'll have 2 copies of a gene. Now these 2 copies of could be the same allele, they could be different alleles. It just depends, and you'll remember that from your genetics where you use the Punnett squares to work out percentages of characteristics. So how are these alleles different then? We know that they can code for the same trait, but a difference in that trait, but what actually makes them different? Now, basically, it comes down to a difference in the base sequence, and we're looking at only 1 or 2 bases being different. It may be the position of the base. Okay, it might be the actual base at that point that's different, so it just depends. Now, these different forms allow for a single trait, such as the ability to see in colour, to have variance. Now, the positions in a gene where more than one variation of a base may be present is what we call an SNP. So an SNP is where there is a difference in bases in a gene. So that's single nucleotide polymorphism. You just need to remember it as SNPs. So how are new alleles formed? So different alleles are formed due to mutation. If there is a change in that base sequence, it is a mutation. So different hair colours are due to mutations in the base sequence. Now, some mutations are harmful. However, some are neutral or can even be beneficial, okay, especially in the case of red hair. Some, however, can be lethal as well. Now, mutations can occur at any point in the stages that we see on the right-hand side there. So during replication, during transcription, during translation. So the change of a base sequence causing that mutation can occur at any time there. As we've said, our mutation could be due to a single nucleotide being left out. So we call that a single base deletion. An extra single base being added in, okay, we call that insertion. We could have base substitution where the wrong base is inserted. For example, adenine is replaced with thymine. Sorry, just let me go back there. Sometimes the infographics are much easier to understand some of these mutations. So when we look at the top, we've got our normal base sequence. Our substitution, okay, you'll see the difference. We've got our thymine, adenine, thymine, adenine, and then we have another thymine, adenine there. So it actually changes the amino acid, okay? So there we add cytosine, guanine in the normal base sequence, and it changed there. So that's that substitution. Insertion. So here we went TTC. All right, we've gone TCC, so we've had an extra base added, which then changes the amino acid sequences along the chain. And we had deletion here where one has been taken out. So the infographics quite often support the message here being given. So it's a good idea to get that understanding from the infographics. So still looking at how these new alleles are formed. So there's several thousand human disorders that are caused by mutations in single genes. Mutations in body cells, so they're those somatic cells, are eliminated when the cell dies. However, mutations that occur in gametes can be passed on to offspring. Alrighty, we're moving on now to look at this whole concept of genome. And we talked about genome back in topic 1. I believe it was topic 1 when we was doing transcription translation. So an organism's genome is all of the genetic information within that organism. It includes both the coding and non-coding sections of DNA. So not all the sections of DNA are used for coding. Okay, there are some other sections there with other uses, but the genome includes all of the DNA. So we're going to move on now, whilst we're thinking of that whole concept of genome, to look at the Human Genome Project. Now, the entire base sequence of human genes was sequenced in the Human Genome Project. So remembering that humans have the same number of genes, that's what makes us a species. Different types of genes, so different alleles, but we've got the same number of genes. So it began in 1990, this Human Genome Project, and was completed in 2003. Today it's possible to identify sequences which are protein producing genes, and there's approximately 23,000 of these in the human genome. We've got non-coding sequences that are not actually transcribed as well. The genome that was sequenced was from one set of chromosomes, just from one person. So it is a human genome, not the human genomes. It's a human genome. The vast majority of base sequences are shared by all humans, okay, about 99.9%. Approximately 0.1% of differences in bases contribute to the diversity of humans, which is pretty unreal because when you think of each human, you think of us as very different. okay, but only 0.1 of differences in bases make up the difference between all humans, which is mind-blowing. Now, part of your course is to consider this whole area of the nature of science. And the area in terms of 3.1 is the development in scientific research to follow improvements in technology. And this is where we look at this whole concept of gene sequences that are used for the sequencing of genes. So let's have a look further. And in your skills and application, we really look at the sequencing of genes further. So techniques used for gene sequencing, and in the background here we actually have a graph of some gene sequencing. We use coloured fluorescent markers to mark the DNA copies, okay? A different colour is used for each of the 4 bases, adenine, thymine, cytosine and guanine. The samples are mixed together, and all the DNA copies are then separated into one lane of a gel according to the number of nucleotides. A laser then scans along the lane to make the markers fluoresce, so make those markers stand out. An optical detector is used to detect the colours of the fluorescence according to the base. So it's now going to pick up which bases are present and in which amounts. A computer then deduces the base sequence from the sequencing of colours, because now you've got these different colours for each base, so you can actually pick up that sequence. Now, that's a very overview of base sequencing and how gene sequencing is done. Let's have a look now at the Sanger technique. So, the DNA is cut into fragments. Copies are made and a primer is added. So, primer allows for the replication. DNA polymerase, remembering that from back in our replication and transcription translation unit. DNA polymerase is used to add 3 nucleotide bases to the fragment, allowing the complementary base pairing rule to occur. Some of the 3 nucleotides have had a fluorescent tag or a label attached to them. These act as DNA terminator, which stops the addition of any further bases. Where the fluorescent base is added in the sequence is totally random. The result is a series of new strands of varying bases ending in one of the 4 fluorescent bases. These strands are then sorted in length using gel electrophoresis, which you would have seen back in year 10. A laser activates the fluorescent markers on the nucleotides, as we just spoke about in our gene sequencing. A sensor hooked up to a computer analyzes the wavelength of light being emitted and determines whether the base is adenine, thymine, cytosine or guanine. So that's what we just spoke about in the previous section. Once the fragment is done, the next fragment of DNA is then sequenced. The final stage is to put all of the fragments in order by lining up any overlapping segments until they match. Alright, so if we move now on to our application skills, we're going to see why some of these techniques are important. Your first section in your application and skills is the causes of sickle cell anemia, including a base substitution mutation, a change to the base sequence of mRNA transcribed from it, and a change to the sequence of polypeptide in hemoglobin. So we're actually going to look at this condition, sickle cell anemia, and see how and why it occurs. So what is sickle cell anemia? Well, when we look at this image here, we can see how in the top pictures, we've got those normal red blood cells. You can see that disc shape, okay? Now, when we look at the lower pictures, we can see the sickle shape of the blood cells with someone that suffers from sickle cell anemia, and we can see how they actually block the flow of blood in the vessels. So sickle cell anemia is caused by a single base substitution in the gene that codes for alpha globin polypeptide in hemoglobin. So we've got this single base substitution. It results in red blood cells becoming sickle-shaped and not carrying oxygen properly. It occurs most frequently in people with an African ancestry, where about 1% suffer from sickle cell anemia and between 10 and 40% are carriers of the allele. Hemoglobin is made of 4 polypeptide chains. 2 are alpha and 2 are beta, bonded together to form the hemoglobin molecule. Now, as we said, this substitution occurs in that alpha globin polypeptide molecule. The beta chain are affected by the sickle cell mutation. Oh, the top thing is saying it's the alpha globin. We're looking at the beta chains here. In a normal allele, okay, and we're calling this HbA, okay, so it's hemoglobin, that's the Hb, A is our normal allele, okay. In the normal allele of a hemoglobin gene, the triplet GAG is found with its coding strand CTC. Okay, so that's straightforward. We've got the opposite there. So GAG is our coding strand, CTC is our sense strand. The strand then used to copy that mRNA into mRNA. The sense strand is transcribed into the mRNA as GAG. Okay, so this is what should happen. This is in a normal allele. During translation, the mRNA GAG results in the amino acid glutamic acid being placed into the chain. So that should all be good. This is our transcription translation occurring here. Now, in the sickle cell anemia mutation, the allele HbS, the adenine base is substituted with thymine on the coding strand. So, the coding strand up here we had GAG, now we've got GTG. So then our sense strand is CAC. This transcribes on our mRNA as GUG. When translated, the amino acid produced is then valine. So we now have a different chain of amino acids. Okay, so normal at the top, we end up with glutamic acid. Sickle at the bottom, we end up with valine, so a different amino acid. Valine has different properties to glutamic acid. The resulting hemoglobin molecule has a completely different shape, as you can see from the diagrams there. Now, the sickle hemoglobin molecules are less soluble. In low oxygen concentration, it deforms the red blood cell to give that sickle shape. Okay. Sickle cells carry less oxygen than normal blood cells because they are a smaller shape. They are stiff, they are sticky, and they tend to block blood flow in the vessels of limbs and organs. Blocked blood flow can cause pain, serious infection, and organ damage, such as enlargement of the spleen, as well as less oxygen carried to cells. Individuals become anemic. Sickle cells are rapidly removed from circulation, leading to a lack of red blood cells, which can then lead to jaundice and kidney problems. Okay, so in individuals that are homozygous for the trait, so we're saying both alleles are the same, they have the disease, sickle cell anemia. Their red blood cells will become sickle-shaped. Less oxygen is carried in those blood cells. The red blood cells do not survive long. They do actually end up bursting. They block blood vessels and capillaries. The person will get circulatory problems, and this may cause pain, and organ failure may be a result. So it is quite a serious condition. Now, in individuals that are heterozygous, so they are carriers for the trait, they may have some sickled cells and some normal cells. The symptoms will not be as severe as someone in homozygote with minor sickling. They will, interestingly enough, be resistant to malaria. And we'll look further into that malaria resistance in class. Okay, your next application, the comparison of the number of genes in humans and other species. So you need to be able to make a comparison between the number of genes that a human has, so we can see there 21,000, okay, compared to other organisms on that list there. A skill to use of a database to determine differences in base sequences of a gene in two species. So we will be doing this further in class, but you need to be able to use online databases such as GenBank to compare the base sequence, the genes of two different organisms. Thank you for watching 3.1. We do go into all of these topics much more in class and also in the coming few videos. Thank you for watching.